The overall goal of this procedure is to rapidly fix intact Drosophila larvae in order to image the morphology of branches and tubes in tracheal terminal cells. This is accomplished by first generating homozygous fluorescent protein-labeled terminal cells in genetically mosaic animals. The second step of the procedure is to collect mosaic larvae and fix them using heat. The third step is imaging labeled terminal cells in fixed animals using GFP expression to observe branches and bright field microscopy to observe tubes. The final step is quantifying the terminal cells' morphologies. Ultimately, results can be used to distinguish and categorize distinct cellular morphologies of wild-type and mutant terminal cells.
The main advantage of this technique over existing techniques such as chemical fixation is that this technique is extremely rapid, so it can be used for high-throughput analysis of tracheal terminal cells.
Generally, individuals new to this method will struggle because picking L3 larvae and quickly fixing them to preserve their morphology requires attention and care.
To make mosaic flies, use the Mark M technique, with females bearing the mutation to be examined. Set up the crosses in pre-lay vials with a smear of fresh yeast paste, and allow the flies to lay eggs for 1 hour at 25 degrees Celsius. Then, transfer the flies from the pre-lay vial to a fresh vial with fresh yeast paste, and allow them to lay for 6 hours at 25 degrees Celsius. After 6 hours, transfer the adults back to pre-lay vials for storage and use in subsequent experiments. Immediately heat shock the collected lay vials in a circulating water bath for 45 minutes at 38 degrees Celsius.